Hello! Okay, so today we're going over lab number one. Lab number one, cells and mitosis, okay? All right, you can follow along with the instructions. We're just gonna go over models and parts of the cell. So let's first go over mitosis. So first here is interphase, okay? The cell is not dividing, nothing much is happening yet. And then we move on to prophase. This is where it actually starts. So prophase, nothing's separated yet, but here you see the centrioles producing the spindles. Yeah, and then here is also prophase. You see more action. We're starting to move the chromosomes. Okay, and then second is a metaphase. And metaphase, we see they're all kind of aligned. A way to remember this one is all the chromosomes are in the middle. And so metaphase, M for middle, okay? So all in the middle. And then we have, next stage is anaphase. And anaphase, they're starting to be torn apart. So the pairs of chromosomes are starting to be torn apart. And that's also a good way to remember. So anaphase, apart. Okay, so the chromosomes are being torn apart. And here we see anaphase at its later stage. So we see the anaphase, it's successfully tore the chromosomes apart from each other. And then here we have telophase. Telophase is where you could almost see the nucleus is obviously already separated in two, or there's two nucleus now, and then starting to just close up and make two brand new cells, and this is the end product, which is daughter cells. So two nucleus, two cells separated. So that's your mitosis model. Okay, next we're going to go over the cells. Cell and their organelles. So first, let's see, we have the nucleus, right? We have the nucleus right here. It's the nucleus. Nuclear envelope is like the whole thing. Okay, nuclear envelope, the nucleus. And then we have here the endoplasmic reticulum, the dark blue. And there's two kinds of endoplasmic reticulum. There's the rough one. You see the little red dots, right? The little red dots. That's ribosomes, and ribosomes are on the rough endoplasmic reticulum. And on the smooth endoplasmic reticulum, there's no ribosomes. Okay, there's no red dots. And so the rough ER, or rough endoplasmic reticulum, they help with transport of proteins. They also store proteins. And the smooth ER, they produce or synthesize lipids and also control lipid metabolism. Next, we have the Golgi Apparatus, and the Golgi Apparatus packages the proteins and it sends them out of the cell in little vesicles. All right, so this is the Golgi Apparatus. And then next we have the lysosomes right here. The lysosomes, they're kind of like gray little balls. Think of them as bombs. So like they destroy, they have a lot of digestive enzymes, and they basically destroy things. Also, another name for them is suicide sacs. Yeah, I call them bombs. Okay, lysosomes. And then we have peroxisomes. They're similar to lysosomes right here. Right here, actually. That's a peroxisome. And one particular thing about them is that most peroxisomes, you find a higher concentration of them in liver and kidney cells. Okay, it's peroxisome. Then next we have your mitochondria. this right here, you probably remember that from high school, A&P. Mitochondria, the powerhouse of the cell, right, generates energy and produces ATP. Then we have your centrioles, this here, these yellow looking things, and they form the mitotic spindle. So these spindles that we saw from mitosis, they help make them. Okay, that's your centrioles. And then you have your cytoskeletal elements. So right here, like number 6. So you have 3. There's a microfilaments, microtubule, intermediate filaments. So they're all part of your cytoskeletal elements. And they all have their different functions. So example, like your microfilament, they're important for moving your cells. So you see here, kind of like, I don't know, little hairs, and they kind of move your cells. Okay, so microfilaments. And then your intermediate filaments, they help resist the any type of pressure or force that's acting on the cell. Okay, so they resist your intermediate filaments. And then your microtubule, they're kind of like the skeleton. They kind of hold up the shape of the cell. All right, that's your microtubules. So here, the thin yellow skeleton of the cell. And that's all for your cell organelles. So this is isotonic balance. There's nothing going in or out of the cell. So the cell stays in normal shape. Or in your demonstration, it says, I think you'll be using an egg, okay? So the egg, nothing will be crossing from the solution to the egg or from the egg to the solution. And this one, this is hypotonic solution. So the solution is hypotonic. And so when it's hypotonic, that means the concentration is going to be a lot higher inside, and so it's going to pull the solution inside to balance the concentration. Okay, so this is hypotonic solution. And this one is the opposite. So this is hypertonic, and hypertonic draws the water or solution from the cell out into the solution because the concentration of solutes is greater in the outside than the inside. So it's going to pull fluid or water from the cell into the solution to balance the concentration. That's it.